Myself Dr. Vivek Kumar, Professor, Himalayan School of Biosciences, Swami Rama Himalayan University, Jolly Grant, Dehradun. I am again with you a new topic on endophyte, that is endophyte plant abiotic stress alleviation. Endophytes, as we discussed before, they are present inside the host plant. They play an essential role in host fitness, nutrient supply and stress tolerance. They are often used in sustainable agriculture as biofertilizers, biopesticides and as inoculant to mitigate the abiotic stresses that could be salinity, drought, cold, metals, pH, etc., etc. And in changing climatic conditions, abiotic stresses create global challenge to achieve optimum crop yields in agriculture production. So, the plants experience stress conditions that involve boosting of their immune system or the overexpression of their defensive genes, overexpression of their defensive redox regulatory systems with increased reactive oxygen species ROS. The rising stress factors overwhelm the natural redox protection system of the plants, leading to massive internal oxidative damage and death of the plant. So, the endophytes are an integral internal partner of the host and mitigate abiotic stresses via modulating local or systemic mechanisms and producing antioxidants to counteract ROS in the host plants. Now, there are different abiotic stresses. So, abiotic stresses are of significant growth and yield constraining aspect of the crop plant. So, if the plants we are cultivating in diverse any abiotic stress, then obviously we are not going to get the optimum yield. Around 90% of cultivable lands are susceptible to one or more abiotic stresses, resulting in about 70% of major food crops yield loss. So, development and yield of crop plants, they are influenced by pH, light, temperature, salinity, drought, floods and heavy metals. And this picture shows there are diverse abiotic stresses and diverse biotic stresses, which we will discuss in the next lecture. In abiotic stresses, salinity, alkalinity, drought, light, ROS, temperature, nutrient deficiency and the environment pollution, they all adversely affect the growth of the host plant. But all these stresses, they are ameliorated by the endophytes, by enzyme SRHU deaminase, antibiotic production, antioxidants, exopolysaccharides, hydrogen production, hydrolytic enzyme, insecticidal, nematicidal nitrogen fixation, pK solubilization and different other mechanisms using with the host plants are able to sustain or overcome the different biotic or abiotic stresses. The assessment based on the combination of crop yield and climate change simulation models have projected more loss in the production of staple crops, such as wheat, maize, rice, resulting in severe concern for global food security and security. So, the application of endophytes in plants has resulted in abiotic stress mitigation by activating host stress response or biosynthesizing the anti-stress biomolecules. Now, salinity and alkalinity, soil salinization with various salts, especially sodium chloride ions, constrain normal plant function, resulting in reduced crop yield. The salt tolerant endophytic microbes have been employed to overcome this problem and considered potential bioinoculants to augment the crop production, growth and yield under saline conditions. So, plants' tolerance to saline stress is related to the antioxidant enzyme elevation as proposed in several reports. Further, the reactive oxygen species ROS scavengers include tocopherol, glutathione and ascorbate. The enzymes included are ascorbate or thiol reliant peroxidase, monodehydroascorbate reductase, superoxide dismutase, catalase and glutathione reductase. So, all these enzymes, they play a significant role in overcoming the diverse abiotic stresses. So, these biomolecules are intricated in ROS removal either by direct process of enzymes such as we discussed before APX, SOD and CAT, or indirect means via regenerating glutathione and ascorbate in the cell. Under salinity stress, osmolytes, they also build up in the organic acids, sugars, glycine, betaine, proline, amino acids, polyamines, they all contribute to the osmotic regulation. So, these osmolytes labeled as compatible solutes are water soluble low molecular weight organic complexes that at higher concentrations are non toxic to the host plant. And if we discuss the drought stress, the drought condition in plants is created either by limited water supply or because plant face a very high rate of transpiration. In typical soil and water conditions, daytime water stress generally happens in most plants during midday and mid-afternoon hours in temperate regions, negatively influences the plant growth rate. So, the drought stress results in decreased germination rate, poor plant growth, decreased photosynthesis, plant cell membrane integrity loss, and the higher reactive oxygen species ROS production. Now, this picture shows the endophytes ameliorating drought and salinity stress. They do so by production of some osmolytes, they do so by induced expression of defensive genes, they do so by production of certain phytohormones. Endophyte Bacillus strain exhibited great 1-aminocyclopropane 1-carboxylate deaminase production. This is an enzyme which breaks the ACC. Endophytic bacteria Micrococcus luteus synthesize indole acetic acid, and Bacillus methylotropic strain produce higher amount of exopolysaccharides. So, the production of this ACCD, production of IAA, production of higher amount of exopolysaccharides, these the secondary metabolites, they play a significant role in the host plant system to overcome the diverse abiotic stresses. And these secondary metabolites ACCD, IAA and exopolysaccharide produced by endophytic bacteria augment Helianthus tuberosus root biomass, enhance lateral roots, produce water produced under water deficit environments. So, it means drought conditions, so production of ACCD, IAA and exopolysaccharide was beneficial to the host plant. So, the inoculation of wheat with endophytic fungi rhizophagus enhance root growth, root biomass and above ground biomass under low water or drought conditions. Root endophyte pseudomonas which have been isolated from a desert plant Algae spressifolia often faces water scarcity. This algae plant faces water scarcity, or we can say it is a drought tolerant plant. When pseudomonas are isolated from this and this pseudomonas were inoculated with wheat, encourage the wheat plant growth under drought endurance. So, the endophyte they synthesize calionic acid, siderophore, spermidine, 1-aminocyclopropane 1-carboxylate deaminase ACCD. All these secondary metabolites might have played a significant role in mitigating the drought stress in the wheat plant. Then there is a temperature stress also. So, temperature enhancement reduces crop production by influencing the molecular, physiological, biochemical and morphological features. The plant growth enhancing endophytic bacterium Bacillus cereus, it has offered a thermotolerance effect on the soybean crop. The inoculation soybean crop or plant with SA1, this Bacillus cereus species, resulted in overexpression of stress responsive GMLAX3, GMAKT, SA1 and decreased ROS generation. So, increased potassium gradients and modulated ABA and oxygen stimuli, all of which are crucial in plant under temperature stress. Endophytic fungi enhance wheat plant heat tolerance, increase root biomass, plant height, enhance grain weight as compared to the control or the uninoculated plant. So, a thermophilic endophytic fungus isolated from the roots of Cullen pelicata, a plant adapted to grow in hot desert, was used as a biocontrol to mediate heat stress resistance in the crop plants. The cucumber inoculation with CPE endophytic fungus enhance the accumulation of antioxidant enzymes, saponins, total sugars, flavonoids and soluble proteins as compared to the non-inoculated cucumber plants. Endophyte Aspergillus niger, when inoculated to sunflower, resulted in enhanced biomass, enhanced plant height and chlorophyll content. So, at a temperature stress of 40 degrees centigrade, Aspergillus niger significantly reduced the lipid peroxidation, concentration and reactive oxygen species. Furthermore, the ROS quenching metabolites like glutathione reductase, superoxidase dismutase, ascorbic acid oxidase, catalase, peroxidase, proline and phenolics were improved. ROS reactive oxygen species, under various stresses, ROS endogenously synthesize signal regulators or molecules generated by the plant organelles such as peroxisomes, chloroplasts and mitochondria. So, these molecules are produced under stress conditions consisting of a group of oxygen molecules that are chemically reactive. For instance, hydroxyl radical, hydrogen peroxide radical, superoxide radical and singlet oxygen. So, ROS overproduction under abiotic stress must be regulated in a homeostatic pool. Surplus ROS leads to oxidative stresses resulting in protein assembly, denaturation, lipid peroxidation and interruption of the nucleotide, and may influence plant physiology, eventually resulting in the plant death. There are several reports which indicate that the role of endophytes in managing ROS under diverse abiotic stresses. The endophytic consortium Rhizobium, Rhodotorula, Acinetobacter, Burkholdia, they reduce damage by ROS under drought conditions, and the consortium also produce an increased amount of indole acetic acid, which all this secondary metabolic production resulted in the overall enhanced growth of the plant. So, under growth conditions, the drought conditions, seed priming of Z maize with Burkholderia phytoferments and Enterobacter mitigated damage caused by the H2O2 production. So, inoculation of Bacillus subtilis with Cicer aretinum under salinity exhibited improved ROS scavenging exudent using the sword podcat enzymes, which resulted in improved Cicer aretinum growth and production as compared to the non-inoculated control plants. Then there is certain environmental pollution. The synergistic relation between endophytes and plants can also play an important role in managing the pollutant degradation in the rhizosphere. Endophytic bacteria could perform more efficiently than rhizospheric bacteria, since endophytes they take part in a course recognized as bioaugmentation, because they are in close contact with the host plant as compared to rhizobacteria. The inoculation Solanum nigrum with Serratia and endophyte lessened growth inhibition in cadmium presence. Similarly, a consortium of endophytes isolated from Agrocystis stimulated Festuca growth in lead polluted soils. Endophytic bacterial strains isolated from Juncus actus exhibited resistance against metal pollutants and other emergent pollutants like your BPA and CIP, SMX. The inoculation of endophytes singly or in consortium enhanced the efficacy of Juncus acutus phytoremediation potential. For the practical bioremediation of ternary effluent, an experiment was planned using an plant endophyte association in a constructed wetland. So, in a vertical flow of constructed wetland, the plant Bracharia associated with 3 bacterial endophyte strains was cultivated, and it was observed that there was reduction in CO2 up to 82%, BO2 up to 94% and metal chromium up to 95% was seen when the plants were inoculated with the endophytes. Another aspect of endophytes is to reduce the plant uptake of pollutants, heavy metals, by binding them extracellularly or intracellularly. Besides, they are also able to modify the chemical structure of the pollutants with less hazardous or non-toxic end products. Therefore, we can say that endophytes they play an important role to overcome the adverse effects of abiotic stresses, resulting in improved plant growth in polluted ecosystems. Finally, we can say that the research work the people have carried out, it recognized the endophytes' various means and processes in defending host plants from various abiotic stresses in sustainable agriculture. So, it is essential to comprehend physiological, genetic and molecular modifications during the interaction of host plants and endophytes. Endophytic plants help plant adapt abiotic challenges such as salt, light, temperature, etc. by sustaining the host plant and enhancing plant growth through direct and indirect beneficial actions. See you in next lecture. Thank you so much.